So I'm Tomas Roslin from the Swedish University of Agricultural Sciences in Uppsala, Sweden. And what I have the pleasure to do is to say what Brian just said, but in more words and with more pictures. Because really I think what's the most exciting is the stage in a project where you already have results, you can learn from them, and you can think of what is it that we are seeing? Where should we be going from here? And the key point here is that what we're doing is not a project that we just perform, we produce the results and it stops. It's a progression. So what we have been presenting today are mostly results from the first phase of the project. But then there are the phases 2 and phases 3. So phase 1 in itself, it's a start, but it's also a goal in itself, of course. Because what we have described is what you can learn in 1 year about Malagasy biodiversity. If you think back on the results that we have seen the first exploration of the amount of insect biomass that is available to the rest of Malagasy nature. For anybody feeding on insects, how much is there to feed on in different parts of the country? And how does that change in time during the year? And how is it distributed in space? How far do you need to move to see entirely new insects? And the answer was just a short step, and everything is new. However, this is just a snapshot. This is 1 year of Malagasy nature. And what we all know is that nature is changing. It's changing in Madagascar, it's changing across the world. We are converting natural habitats to man-made habitats. We are appropriating the main parts of this globe, and we are heating it at the same time. And this is not going unnoticed by the insects or anybody else. We've seen that insects across the world are already declining. This is a map of every time series, every public time series that there is on insects from different parts of the world. And the redder the color, the steeper the decline of insects in this part of the world. We see an overall red tone on this map, on a little blue which would signal an increase. And for Africa and for Madagascar in particular, we see nothing at all. Because there are no time series of more than 10 years. Because nobody started them 10 years ago. And that is why we need to advance from phase 1. We should be moving into step 2 and step 3. And we're doing that because phase 2 has already started. Phase 2 does 2 things. It takes the first year of data that we've heard about and extends that. Does the same thing in the same sites, at the same sites, for 6 more years. And then it does another thing, and that is that it expands beyond insects to the other organism groups. So if today Laura was showing this map of how biomass was distributed in 2019-2020, what we are already doing is to follow up on that map and to generate new maps for the next 6 years. And here I've simply copied this map for all the future years. I will be surprised if that proves a fact, if the maps remain unchanged over this time period. The other thing we can do is to take Laura's second picture of how biomass is distributed across the year. And we extend that. And we look at whether or not indeed we are seeing changes in the amplitude of these fluctuations and the timing of these fluctuations across Madagascar. And then we're asking, how representative are the insects of the rest of the biodiversity? Because even if we hate to admit it, there are other creatures out there beyond insects. Insects are particularly diverse, but so are fungi. We know extremely little about fungi. So are microbes that the author was talking about. And these guys are not particularly diverse, I must admit. But most of what we know of the world and about Madagascar comes from birds and mammals. So shouldn't we be asking, are the few things that we know about nature from the few well-studied groups, are they actually matched in the more diverse groups? And to do so, we must study them in the same places using similar techniques to get to a comparison. And that is what we're doing in phase 2. We go for, just as in the IBA, we use the strictly standardized high throughput method for quantifying insect communities. We do the same for these other groups. So for fungi, we actually suck samples of air and look for fungal spores, which is an extremely efficient way of characterizing fungal diversity. For microbes, we take litter samples and sequence them. And for vertebrates, we look for them using camera traps like this one here. And we record their sounds using audio recorders like this one here, which are actually nowadays fairly cheap and easy to operate. And we can introduce you to those in further detail during the break and after the break. Now, all of this we do at each of these sites, which are, to the most part, perfectly overlapping with the IBA sites. So we're really continuing in the same sites, the insect data, plus expanding to the other organism groups. And then up here in the northeast, where biodiversity is really at its highest, we do intensive sampling within that region. And to get the global comparison for these data, we're doing the same things in all of these black dots across the world. So Madagascar has the most intensive sampling, but we do the same and generate exactly the same data that we can compare to from across the world. And just to show that there's much to be found in these data, I would like to show a very first result from fungi, because I think that's really, really one of the most promising areas. And just like the insect, this is an exceptionally diverse group of which we know virtually nothing. So here, remember that we're sampling now at all of the black sites. Here, I will just show you results from one site, sampled from 1 year, up here, using this kind of suction device, and then sequencing a sample which is collected in a tube this large, and when you look at it, you can't see anything at all because the spores are so small. Of that invisible diversity, we actually find that in this site, we can find of the OTUs or species-like entities that we're finding, we can be next to certain about the fact that almost 60% of them have never been seen by anybody anywhere before. And another about 20%, it's likely that they haven't been seen before. So some 80 to 90% of what we're finding in this simple suction device in one site of Madagascar is new to science. Now that's diversity, in a tube that you can't see any kind of contents in. Now, this was phase 2. But of course we shouldn't stop after 6 years. As Brian was saying, what we really want to do is to continue this forever. Why should we stop? There's no point when we know what Malagasy nature and biodiversity looks like, and if we think so, we're wrong because it's changing all the time. So we need a long-term monitoring program all across Madagascar and all across Malagasy biodiversity. So if after IBA and life plan we can draw these maps, then what we would really like to see is the day when we can draw all of these maps. And I bet you my annual salary that they will not look the same at that point in time. Once we have this data, this is what we can do. We can feel the temperature of Malagasy nature, and we can say, these are the different trends in the different organism groups that we are seeing. What action can we take? How well are our reserves protecting species? How are they doing? Now, how do we make this happen beyond using nice words? What we have been saying, and what I will say once more, is that the first thing we need to do is to act now. The data that we do not collect now cannot be collected for year 2023 in 2030. This is our chance to capture the state of Malagasy nature now, and we need to establish what is there now to have a point to refer to. What we're doing in this phase 1, the IBA, phase 2 life plan that we've been talking about, is actually figuring out the means. Like, what can actually be done? How should we do it? And how much does it cost? How realistic is it? The other thing that we are doing is to, first we create the methods, then we teach everybody else to use them. What use is a method that you develop for yourself and keep to yourself? It's just senseless. The whole rationale of developing a method is because you want other people to use it to make sense of nature. So a key part of phases 1 and 2 is to teach anybody interested here in Madagascar to know how to generate the samples, prepare them for analysis, to conduct the action analysis, and to make sense of them, to see where nature is going. And as Brian was strongly emphasizing, and as I will emphasize once more, the ultimate objective is, of course, that this should be done to the maximal part possible in Madagascar by the Malagasy people. Otherwise, we would have failed. And if there is anything that I want you to remember of this day, is that in terms of generating a Madagascar biodiversity monitoring program, we can make it happen. We have just shown that, and we are making it happen. It's not empty talk. We have shown you we can do it. The aim here is to generate the methods and disseminate them. And the third key point is represented by all of these logos, and that is the only way we can do this is together. Because that's how the project has been run so far. I mean, who did all of the sampling? Who gave all of the permits? Who should be learning about what we're finding? It's all these, it's all of you. And that's why I would like to end by really, really thanking you. And I would also like to thank the more than 100 Malagasy local assistants who have actually generated every piece of the data that we have been talking about here today. Thank you very much.